Welcome back to the another video for chapter 14, that is respiration in plants. In this video we are going to study about the aerobic respiration. So first recapitulate what is aerobic respiration. It is the process that leads to the complete oxidation of organic substances in the presence of oxygen and releases carbon dioxide and water with the expenditure of large amount of energy. This type of respiration is common in case of higher organisms. The process of respiration takes place in mitochondria, and the final product of glycolysis, that is pyruvate, is transported from the cytoplasm to the mitochondria. Let us consider the crucial events that are going to happen in case of aerobic respiration. That is the complete oxidation of pyruvate by stepwise removal of all hydrogen atoms, leaving 3 molecules of carbon dioxide, which is occurring in case of Krebs cycle. The cycle is known as Krebs cycle because it is elucidated by the scientist Hans Krebs. This process takes place in the matrix of mitochondria. In the another event, there is passing on of electrons that are removed as a part of hydrogen molecule to molecular oxygen with the synthesis of ATP, which occurs in electron transport system. This process is located on the inner membrane of mitochondria. Now the pyruvate, which is formed in the glycolysis or the glycolytic catabolism of carbohydrate in the cytosol, enters into mitochondrial matrix that undergoes oxidative decarboxylation. With the help of enzyme, the set of reaction is catalyzed by the enzyme pyruvic dehydrogenase. This pyruvic dehydrogenase requires the participation of coenzyme A and NAD+, thereby making the reaction pyruvic acid plus coenzyme A plus NAD+ in the presence of magnesium as a catalyst and the enzyme pyruvate dehydrogenase results into the formation of acetyl coenzyme A, carbon dioxide, and reduction of NAD+ to NADH to H+, wherein the acetyl coenzyme A that enters a cyclic pathway, that is TCA cycle or Krebs cycle. Now after the respiration, we will be going further towards the Krebs cycle or TCA cycle. Now coming to TCA cycle, that is tricarboxylic cycle or the Krebs cycle. As we have already discussed that TCA cycle or Krebs cycle takes place in the matrix of mitochondria. Now the question comes, why this cycle is called as TCA cycle? The full form of TCA is tricarboxylic cycle. That means in this cycle there is consecutively 3 carboxylation reaction occurs, and because of these 3 consecutive carboxylation reaction, the process is called as tricarboxylic cycle. The cycle starts with the condensation of acetyl group with oxaloacetic acid and water, which leads to the formation of citric acid. The reaction is catalyzed by the enzyme citrate synthase and a molecule of coenzyme A is released. The citrate is then isomerized to isocitrate, which is followed by 2 successive carboxylation and leads to the formation of alpha ketoglutaric acid and succinyl coenzyme A or succinic acid. On the formation from citric acid to succinic acid, there is 2 successive carboxylation occurs and NAD+ is reduced to NADH and NADA plus is reduced to NADH, leading to the formation of 3 NADH. During decarboxylation from alpha ketoglutaric acid to succinic acid, there is formation of GTP, that is guanosine triphosphate, which is an energy rich compound, is converted, and with this there is a simultaneous synthesis of ATP as well. From succinic acid, regeneration process is carried out, resulting into the formation of 1 FADH2 and malic acid is formed. From malic acid, it is then converted to oxaloacetic acid with the reduction of NAD plus to NADH. Thereby the reaction is taken out, or it is carried out with the following reaction, with the following substrates as well: pyruvic acid plus 4 NAD plus FAD plus H2O plus ADP plus Pi in the mitochondrial matrix results into 3 molecules of carbon dioxide, 4 NADH molecules, 4 H plus ions, FADH2 and 1 ATP synthesis occurs. It is counted as NADH is counted as 1, 2, 3 and 4, thereby making 4 NADH molecules, 1 FADH2 molecule, 1 GTP, or with the GTP there is a synthesis of ATP as well. Thereby, hence, the reaction for TCA cycle in the mitochondrial matrix is as follows. Thus, we can say that in TCA cycle, the energy is stored in the form of NADH and FADH2 and ATP. Next, we will be talking about electron transport system and oxidative phosphorylation. Now we are going to discuss about electron transport system. The electron transport system takes place in the inner mitochondrial membrane of mitochondria, and the hydrogen ions are passed on to intermembrane spaces, and in the matrix the TCA cycle has been accomplished before the electron transport system. Now, in this system, NADH is reduced to NAD plus in the TCA cycle, is oxidized through electron transport system, and the electrons are passed on to oxygen, thereby resulting in the formation of water. This metabolic pathway through which the electrons are passed on from one carrier to another carrier is called as electron transport system, and this electron transport system is accomplished by 4 complexes: 1, 2, 3 and 4. These are the 4 complexes by which the process of electron transport system is accomplished. Now, NADH2 or NADH is reduced to NAD plus in the matrix of mitochondrial membrane during TCA cycle, is then produces the electrons that are produced from NADH in the mitochondrial matrix during TCA cycle are oxidized by the enzyme NADH hydrogenase in the complex 1, and the electrons are transferred to ubiquinone located at the inner membrane. The ubiquinone then receives the reducing equivalent via FADH2 and is generated during oxidation of succinate in the citric acid cycle and results in the oxidation with the transfer of electrons. Now, the reduced ubiquinone is then oxidized with the transfer of electrons to cytochrome B and cytochrome C complex, and cytochrome C is a small protein that is attached to the outer surface of the inner membrane, thereby acting as a mobile carrier for the transfer of electrons between complex 3 and complex 4. This complex 4 refers to as cytochrome C, and cytochrome C oxidase complex containing cytochrome A and cytochrome 3 has 2 copper centers. When the electrons are passed on from one carrier to another via complex 1 to complex 4 in the electron transport chain, they are coupled to ATP synthase for the production of ATP and inorganic phosphate. The number of ATP molecules synthesized depend on the nature of the electron donor, and the oxidation of one molecule of NADH give rise to 3 molecules of ATP, while one FADH2 results in the production of 2 molecules of ATP. As seen here, that 1 molecule of NADH results into the formation of 3 molecules of ATP, and 1 FADH2 results in the formation of 2 molecules of ATP. The complexes that are formed in the electron transport system is first complex 1 NADH dehydrogenase, complex 2 succinate dehydrogenase, complex 3 that is cytochrome BC1, complex 4 that is cytochrome A2A3, and complex 5 ATP synthase. Here oxygen plays a vital role in the removal of electrons and hydrogen ion, and finally this helps in the production of water molecule or hydrogen atom. Thus, oxidative photophosphorylation or oxidative phosphorylation means the process that accounts for high ATP production, or the yield accounts for oxidative phosphorylation that occurs after electron transport system. This yield of ATP results into oxidative photophosphorylation. Thank you for watching this video. Hope you have liked this video. Thank you so much.